as a whole does. And then we'll try to go deeper into how it acts. Okay, all through one single flow chart, like I've been doing in all of the endocrine chapters. Okay, so to start off, I'm going to draw your pancreas. Okay, so pancreas, the endocrine component of it, has multiple different parts to it. That is your alpha, beta, delta, your F cell. Okay, so I'll just take one part, which is a major part of it. We'll start off with your beta cell. So the first thing is your beta cell, which is around 60% of your endocrine pancreatic cells. Okay, majority is beta cells. What does beta cell do? So in the beta cell is where your insulin is being released. Insulin is first in its pre-pro form. Okay, this pre-pro insulin is cleaved into a pro form. That pro insulin again is cleaved to finally give you insulin. So this initial step of pre-pro to pro is happening in your rough endoplasmic reticulum. And then in your trans Golgi, your next steps happen. Okay, and insulin is released. Now this insulin which is released, it is actually released in secretory vesicles and stored over there. So this is there in your secretory vesicle. And inside that, we have your insulin. Now, one thing I want you to understand is in the pre-pro form, as it is cleaving and going on, you have one A component, a B component, and a C. Different chains are there. Okay. This A component, A chain is around 30 amino acid. C component is around 31 amino acid. B component is around 21 amino acid. Okay. In the pre-form, there is another small area, 25 amino acid area, which is there, which is first cut off. And then your pro-form is what you see like this. And finally, this C peptide is cut away and you get your insulin. So in these secretory vesicles, you have to understand your insulin and your C peptide are in 1:1 ratio. So it's not only insulin that is being released from these secretory vesicles, but also C peptide, and it is in your 1:1 ratio. Okay, so this is a type of peptide hormone. Okay, so since it is a type of a peptide hormone, regulated exocytosis is what happens. It waits for some sort of signal, and till then, preformed vesicles in it is stored and kept, and when the signal is rising, it results in release of the contents. So contents will be released. Okay, but how does it detect? What does it detect? That is what we have to understand. So this beta cell in the pancreas, what does it detect? Whenever there is glucose, excess glucose which is present, that triggers insulin. Okay, so when a lot of glucose is present, that is when insulin is being released. Quite contrary to that, the counter-regulatory hormones, all the other hormones we started talking of with in the previous lectures, they are released when glucose is less. Okay, so here what happens is when glucose is there sufficiently, this glucose is coming in through a particular transport. This is what you call GLUT2. Remember this is different from the usual GLUTs that we talk about in this chapter. Okay, so this is glued to. It does not need insulin. Usually, for glucose to come into cells, you need insulin. But this is one place where without insulin also, through GLUT2 receptors, glucose is going to come in. Okay, so through glucose comes in through GLUT2 receptors. Now, glucose is inside. Glucose now again gets utilized for your glycolysis. After glycolysis, your TCA cycle, and you finally have ATP being formed. So at rest, when glucose is not there, you have a particular transporter over here, which is a potassium ion channel. So this potassium ion channel, what does it generally do? It takes potassium outside. Potassium efflux. When there is potassium efflux, positive ions are reducing inside, going more towards hyperpolarization. Okay, so it's keeping this beta cell inert at rest normally. Now, once glucose comes in, you use glucose for glycolysis and you form ATP. Then what happens is your ATP will go and inhibit this channel. So this is an ATP sensitive potassium channel, which gets inhibited when glucose is present. Okay. So now that this is inhibited, what is the scenario? More potassium ions keep accumulating inside instead of going outside. So inside becomes more positive, which means the threshold for depolarizing some other channel will be much more easier. Okay, what is that other channel? That is your calcium. There's another channel where your calcium comes inside. Now we know that calcium coming inside can do a lot of things in the body. Muscle contraction being one among them. But even for your exocytosis, you need calcium. So that calcium, which has just now entered inside, will make sure that these secretory vesicles will give out insulin. So that is how finally your insulin is being released. So how does that happen? In the beta cell, glucose comes in through GLUT2 receptors that is used for glycolysis. And once ATP is formed, that ATP inhibits your potassium channels. Okay, that will result in increase in positive ions inside, resulting in calcium influx. That calcium, which is coming inside, will lead to the preformed secretory vesicles to be extruded outside. Okay, and what are the different components that are coming out from this? One is your insulin. So we have your insulin, which is there. And we also saw that C-peptide is also there. You have to also understand beta cells, along with insulin and C-peptide, there's another component which is being released. That is your myelin. Okay, so three components are released from your beta. Myelin, insulin, C-peptide. Okay. Next, we'll go to the next cell, which is your alpha. Okay, but before that, we just quickly try to understand what does myelin do, what does insulin do, and what is the functionality of C-peptide. And then we'll go to it in detail later. Okay, so what does insulin do? Problem now was a lot of glucose is available, which means all the body cells in the body can use glucose. Okay, so hyperglycemia is always a problem. In the long run, if too much glucose is there in the blood, that will result in free radical, and that will result in damage in the future. That is what happens in diabetes. Okay, so hyperglycemia is not a good thing. So glucose, which is there in excess now in the blood, should be reduced. And since glucose is there in plenty, glucose can be used for energy. Okay, now if you look at your body, there are few cells that require only glucose, cannot use fat or protein. For example, yes, nerve cells in the brain, germinal epithelium. Okay, so certain cells that require only your glucose, usually they're the ones that use glucose. But when a lot of glucose is there, everybody else can use. Who gives a signal for that? Insulin. Otherwise, when not enough glucose is there, everybody is restricted from using glucose. Only those special people can actually take glucose. Okay, so now insulin, what does it do? It tries to reduce blood glucose. How does it achieve it? It achieves that by one, increasing your glucose uptake. So glucose uptake into the cells is increased and glucose utilization. So not only uptake, the utilization for glucose for energy is also increased. Okay. Now excess glucose which is there stored as glycogen. Okay, so whatever is used will get used, the rest of it is stored as glycogen. Okay, and since a lot of glucose is available, we don't have to break fat or protein right now. This generally favors lipogenesis. And actually, extra glucose can also be converted to fat, actually. And it favors protein synthesis. And the whole point of this is to try to reduce glucose. So anything that generally increases glucose will be inhibited. One example is your gluconeogenesis. So gluconeogenesis will be inhibited. Okay. So these are various functions of your insulin in a nutshell. Now, if you look at amylin, surprisingly, amylin also has a similar sort of function at the top. That is, its core function is to reduce the blood glucose. But how it does it is completely different. So this is what insulin does. How does amylin act? Amylin decreases blood glucose, one, by reducing food intake. Okay, so while eating food, amylin, glucose is there, and now amylin also released, that will inhibit the food intake. Then what it does is, so what will happen? The amount of glucose that is being taken is reduced. Okay, then your gastric emptying is, the time for gastric emptying is increased. Okay, so delay in gastric emptying, which means the amount of glucose coming in for absorption is also reduced. Okay, so it decreases the food intake, gastric emptying delays it, and glucose absorption is reduced hence. And also, all the counter-regulatory hormones we were talking about before, which serves the exact opposite function. What does counter-regulatory hormones do? They generally try to increase the blood glucose. So counter-regulatory hormone release is decreased. So in these ways, amylin now decreases your blood glucose. Okay, there are some studies that also say that amylin has an inhibitory role on insulin also. Okay, so again, this is something you don't really find in textbooks. Amylin, why it is released and what is its functionality. Okay, so now what do you have? We have amylin, we have insulin, both try to decrease the blood glucose. What is C-peptide? C-peptide per se, we haven't really found functionality for it yet. Okay, it's just a broken down fragment of its proform, which is just coming along with it. But you can use it. How? As an investigative module. For example, imagine the person is diabetic and you're giving insulin from outside. Now you need to understand you're giving insulin from outside. On top of that, if you want to understand how much insulin is innately being formed from this person. Okay, so to measure that, if you measure how much C-peptide is there, because it is in 1:1 molar ratio, you can understand how much of insulin is provided from outside, how much of insulin is this person able to form by themselves. So like that, C-peptide can be used as a investigative module. Okay, so these are all the things that are released from your beta cells. Do you have any doubts so far? If you do, let me know. Okay, so next, beyond beta cells, we'll go to the next cells. Okay, so that is your alpha. Your alpha cells are the next majority, around 25%. Okay, so the alpha cells have a pro glucagon. So this is a large peptide which is there. So this large peptide has various components in it. This GRPP is glycine related polypeptide. Then there is a component of glucagon. There is a component of GLP-1, another component of GLP-2. Okay, and some signal peptides and all, not mentioning over here. So imagine a large fragment like this, a pre-pro-glucagon. Now this is cut in various ways to get various products that we require. Okay, it can be cut to give GRPP, okay, glycine related polypeptide, or it can give glucagon, or it can give GLP-1 or GLP-2, or it can give something called glycine. This component will become glycine. Or from this component, you can also get what we call oxyntomodulin. Okay, and this component also together sometimes can come out, which is MPGF. So what I'm trying to say is there are various components that come from alpha. So like we saw amylin, insulin, C-peptide coming from here, from alpha cells, a whole bunch of products come. Okay, it comes from one single structure, which can get split up into various ways. And the core component of that is your glucagon. Okay, the rest of it I'll tell you. So what does glucagon do? Glucagon does the exact opposite. Okay, it's part of your counter regulatory system. But its major function is one, this gluconeogenesis. So that way it tries to increase the blood glucose. Okay, and another way it can increase is glycogenolysis. So it's as if whatever insulin did to store the excess glucose as glycogen can be now broken down by your glucagon. Okay, so these two are the most important functions of your glucagon. Apart from that, of course, it does result in increase in lipolysis, decrease in lipogenesis, and decrease in protein synthesis. These are the functions are also seen, but most important one is this gluconeogenesis, glycogenolysis. That is what glucagon does. Okay. Now, apart from glucagon, we have glycantin. Glycantin also has glucagon-like activity. Okay, so glycantin has glucagon-like activity. What about your GLP-1, GLP-2? Well, these are usually released when you eat food. Okay, in response to food, these products are released usually. Okay, so these GLP-1 and 2 can in fact go and stimulate insulin secretion from the beta cells. Okay, so does MPGF, the same thing. Oxyntomodulin decreases the food intake. It acts on the brain and directly decreases the food intake. And GRPP is one thing that increases acid secretion, increases gastric acid secretion. Okay, so these are all the components which are released from alpha cell and the various functionalities of them. Okay, so pre-pro-glucagon, your glycantin-related polypeptide, which is increasing gastric acid secretion. Okay, oxyntomodulin decreases food intake. You have your glycantin, which has glucagon-like activity, and GLP-1 and 2 has insulin secretory activity. It stimulates beta cells to release insulin. Okay, glucagon, what does it do? Gluconeogenesis, glycogenolysis. This is the main thing that you need to know about glucagon. Apart from this, we have the next cell, which is your delta cell. So the next cell is your delta cell. That comes to around 10%. So that is where your somatostatin is released. The variant of somatostatin released here is 14. In growth hormone, we had seen that the variant which is released is 28 in the brain. Okay, over here in the GIT, it is somatostatin 40. What does it do now? This somatostatin, just imagine if you look at the function of insulin and glucagon, they're like small kids, siblings fighting against each other. Insulin does something, glucagon does the quite opposite thing. They're always fighting against each other. And when it's time, finally the mother has to come and put a stop to the fight. So that is what somatostatin does. Somatostatin now comes, it tells the alpha cell, enough. Don't release glucagon. It also goes and tells your beta cell, enough. You don't release insulin either. Okay, so it has the capability to inhibit both the alpha cell and the beta cell. Apart from that, it also has some other functions in your GIT. Okay, so in the GIT, it generally reduces the motility. It reduces the secretory abilities as well as reduces absorption. So all of this, it's what it does in GIT. Okay, so it's generally an inhibitory activity. Okay. Now, if you look at the percentage, 60%, 25%, and 10%, it doesn't add up. So there are still some other cells which are there. Okay, one example of that is your F cell, also called PP cell. What does it release? It releases pancreatic polypeptide. Now, what does this do? Pancreatic polypeptide, this acts more on your liver. Okay, the pancreatic polypeptide, it acts more. Just a minute. Okay, just give me a minute. My pen's nib just fell out. I'll try replacing it. Okay, so sorry. Okay, so I'll try teaching with my laptop only. So sorry, I'll have to remove the webcam because of that. Okay, so I'll just continue from where we left off. So we were talking about your pancreatic polypeptide. So your pancreatic polypeptide, what it does is it acts on your hepatic cell. Okay, so in the liver, what it does is it sensitizes. It sensitizes your liver to the action of insulin. Okay, so it sensitizes your liver to the action of your insulin. How does it do that? By increasing your insulin receptors. Okay, so now your hepatic cell has been sensitized for the action of insulin by increasing insulin receptors. Also, another thing it does is there is a hormone called resistin. This resistin generally increases your hepatic insulin resistance. Okay, so that ability of this to inhibit your hepatic insulin resistance is something that happens. Okay, so pancreatic polypeptide acts in two ways. One, it sensitizes your liver for the activity of insulin. Two, it acts on resistin and inhibits resistin. What does resistin normally do? It increases hepatic insulin resistance. Okay, so now we talked about all the different components which are there. We talked about your beta cell, we talked about your alpha cell, we talked about your delta cell, we talked about your F cell. Okay, beta cell was releasing a myelin, insulin, C-peptide, alpha cell, GRPP, glucagon, glycine, oxyntomodulin, then your GLP-1, 2, MPGF. Delta cell, somatostatin. F cells, pancreatic polypeptide. And what does all of this do? Okay, so now that we cover this, finally, your insulin is released. So this insulin, which is released now, is going into your blood circulation. So some key points that you need to understand, which is going to be helpful in your clinical practice also in the future. So insulin generally, it has a half-life of around 6 minutes. Okay. Although it has a half-life of 6 minutes, its peak of activity is around 30 minutes to 1 hour. Okay, that is at the peak of activity. But there are different activities of your insulin. It has rapid functions, like wherever ions are taken in, glucose is taken in, all of that is a rapid activity. Okay, they have also intermediate activity and then long-term activity. So rapid is the intermediate, is the long-term activity is there. All that is there when you talk about your insulin. Okay, now comes the point I was telling you, which is very important. Insulin has a basal, when you talk about the secretion of insulin, it has a basal secretion of around 1 unit per hour. So the basal rate at which insulin is being secreted is around 1 unit per hour. Okay, so in a day, how much would it come to? It should ideally come to around 24. But if we see in a day, on an average, it comes to around 40 units per day. So how does that happen? It doesn't add up. So this is where your stimulated release is happening. That means every time you eat food, there is a peak in insulin. Okay, so apart from the basal, on top of that, there is a 5 to 10 times the basal levels which are released each time you eat. So throughout a day, when you look at it, it's not 24 instead, although it is 1 unit per hour for a basal level. In a day, because of the 5 to 10 times increase during stimulation, it comes to around 40 units per day. So these values I want you to remember. Because when you give insulin to patients for treatment, you have to understand this concept. Okay, and based on this, you decide how much units of insulin you need to give to a patient. Okay, so once that is done, we'll now go on to understand how exactly insulin acts. Okay, so we saw that one insulin has a half-life of 6 minutes, but you see a peak of activity much longer. Okay, so there is a short way to understand this. There is a complicated, long way to understand it. So I'll just try to teach you the basic version first. Okay, and after that, if you do require the complicated version, I'll be more than happy to help you out with that. Okay, so now we have your insulin which has been released, which is going in the circulation. It does not need any binding proteins generally because It is a water soluble it's a polypeptide. Okay. Now I'm gonna draw a cell with this denoting the membrane. Okay. Now if you look at it, it has extracellularly two alpha components, and transmembrane it has two beta component. So this is the structure of insulin. Insulin receptor. Okay. Now your insulin is gonna come and bind to this. So when it comes and binds to this, you have to understand what type of a receptor this is. This is a membrane receptor without doubt, but is an enzyme linked membrane receptor. So this membrane linked enzyme receptor has some enzymatic activity, not coupled to GPCR and all that. Instead, enzyme. What is the enzymatic activity this has? It has an intrinsic activity. Intrinsic tyrosine kinase. Okay, so your insulin receptor, the beta component, has an intrinsic tyrosine kinase activity. What does it mean? Kinase phosphorylates things. Where does it phosphorylate? Tyrosine residues. So, in a very short version, I can just tell you there is a group of structures which we term insulin receptor substrate. Okay, this is just a simplified short form. Insulin receptor substrate 1. Okay, so this insulin receptor substrate 1 has various tyrosine residues, which is now going to get phosphorylated. Once phosphorylated, this can act in two ways. Okay, this is similar if you remember when we talked about growth hormone. This is similar to your insulin-like growth factors also. Okay, even the receptor has very similar homology actually. Okay, now insulin receptor substrate, once it is active, it acts by two mechanisms. One, through PI3K, phosphoinositide 3 kinase, or another mechanism which is mitogen activated protein kinase. So whatever metabolic functions that you see because of your insulin, that is through your PI3K. And through your MAPK, that is MAP mitogen associated protein kinase, that is going to be your growth promoting activity. Your growth promoting activity is through mitogen activated protein kinase, and PI3K, your metabolic activity. So what are the metabolic activity? That will come to. Okay. So just continue drawing this in. Okay. So what was the problem again? We started with excess of glucose, which is there available outside. So now insulin came and acted. What is the main purpose? To take the glucose inside. So what does it have to do? Here, there are vesicles which contain particular transporters, which is GLUT4. So remember earlier, we were talking about GLUT2. But here, by the activity of your insulin, now what is going to happen? Your GLUT2 will be stimulated. It'll go into the membrane. Okay. And a lot of your GLUT4 will now be put into the membrane. As a result of which, now glucose comes inside. Now, once glucose comes inside, again, that glucose should be utilized for your glycolysis. That is also done by your insulin. Okay, so two things this insulin does. One, by producing and putting more GLUT4 onto the membrane, your glucose uptake into peripheral cells is increased. Okay, another thing it does is it acts on a particular enzyme, phosphofructokinase, and stimulates it to increase your glycolysis. To stimulate your glycolysis. That means glucose is first of all taken into a cell, and that glucose is utilized now for energy. So glucose becomes the major source of energy right now. Okay. Now, apart from this, we have your liver. So whatever excess glucose is there, which is utilized for energy. Okay, we don't need too much energy. How much ever your metabolic requirements need, that much is utilized. The remaining glucose which is there, which has gone into the cell, that glucose is now converted to your glycogen. The thing about the glycogen in your liver is, when needed, later, like for example, glucagon acting, your glycogen can be converted to glucose and glucose can be given to the rest of the body. Okay, so glucose can be given to the rest of the body that way. So that is it from your glycogen. Now, apart from this, in your liver, amino acids, from there, proteins are made, fatty acid, lipids are also made. So in your liver, what does it do? Again, uptake of glucose is also favored in the liver. Conversion of glucose to glycogen is done in the liver. Okay, amino acids, from there, proteins are built, lipids are also built. So this too happens in the liver. Because here, amino acid and free fatty acids that may be coming to the liver did not need to go into gluconeogenesis. Okay, so this is what it does to your carbohydrate metabolism. So generally, it is anabolic. Then we have your lipids. Okay, so in your lipids, you have lipoproteins which are there. Attached to these lipoproteins, you have your triglycerides. So we have your LDL, VLDL, HDL, all of these lipoproteins are there. With these lipoproteins, generally, your cholesterol, triglyceride, all that is carried in the body. Okay. Now, all of this, if it is going to your adipose, okay, this triglyceride, which is here, can get broken down to give free fatty acid. That free fatty acid can now be taken inside. And once inside, this free fatty acid will be converted to triglyceride and stored. So imagine triglyceride cannot be directly taken into adipose. Instead, what happens is the triglyceride which is there outside attached to the lipoprotein will be split into free fatty acid. Then it is taken into this adipose. And then it is converted to triglyceride and stored there. So for this, we have a particular hormone, not hormone, enzyme, lipoprotein lipase. So this lipoprotein lipase, as the name suggests, it acts at the level of lipoprotein, splits, it's a lipase, splits triglyceride to fatty acid. But although this is a lipase, ultimately what does it do? It makes sure that the fatty acid goes into the adipose and then gets refined and converted to triglyceride and kept there. Okay, this is opposed to another hormone we talked about before, which splits this into fatty acid. That is what we call hormone sensitive lipase, which is inhibited by your insulin. All the counter-regulatory hormones increases. So what does your insulin do over here? One, it activates the lipoprotein lipase. Two, it inhibits the hormone sensitive lipase. So as a result of this, what is happening? Lipogenesis. And also you have to understand, glucose transport into adipose is also increased. So when there is excess glucose that you continue taking and you're not doing enough activity to burn it down through glycolysis, either it will be stored as glycogen or the glucose by itself can be converted to glycerol 3-phosphate and then can give and be stored as triglyceride. So this can also happen. Okay, so glucose can be converted to triglyceride and stored. This is also something insulin does. Now coming to your muscles. Okay, muscles majorly is where your protein synthesis needs to take place. Okay, so here what happens? Amino acid coming in. That amino acid converted to protein. This is stimulated. On the other hand, your conversion of protein catabolism to amino acid, this is decreased. So this happens in muscle. On top of that, like we talked about earlier, again, glucose also comes. And glucose is used, glycolysis, and energy requirement is met. Okay. And also the excess glucose which is there gets converted to glycogen, just like we saw earlier. But there's a small difference. This glycogen which is here, this cannot, how it came from the liver, go back into the circulation and increase the blood glucose. But when requirement is there and energy is less, this glycogen can now be utilized to give energy later. So that is something a little different in your muscle. Okay, now apart from all of this, one small other component that you need to know is in all the cells, insulin makes sure that potassium comes into the cell. Okay, this is something we need to learn because when we think about treatment for certain diseases like diabetic ketoacidosis, this comes into play. Okay, so what do we have here? Insulin, it acts on the insulin receptor, it's an enzyme linked receptor. Okay, that is intrinsic tyrosine kinase. It has an alpha component and a beta component. Beta component has an intrinsic tyrosine kinase activity. It acts on insulin receptor substrate 1. Okay, it gets phosphorylated. It has two mechanisms. One, PI3K, through which metabolic functions happen. Or it has a mitogen activated protein kinase pathway, through which growth is promoted. Okay, so what are the metabolic changes that happen? One, glucose uptake into the cell. Two, glycolysis is promoted, use of glucose for energy. Three, glucose is converted to glycogen and stored. And in the liver, amino acid and protein synthesis and lipid synthesis is favored. Then lipoprotein lipase in the adipose tissue is stimulated, resulting in lipogenesis and inhibition of hormone sensitive lipase. Again, from glucose, again triglycerides can be formed. In the muscle, amino acid from their protein synthesis is increased and breakdown catabolism of it is decreased. Okay, here also energy is utilized from your glucose. Glycogen is stored, which can later be used by the muscle when required. Okay, apart from all of this, potassium influx is also happening. Okay, so this basically covers at your level what you need to know about insulin. In the next chapter, in the next class, next week, I'll be talking about how to apply this, the various components of diabetes mellitus, its complications, different types, including ketoacidosis and all that, because this is a very important question. Okay, so I'll cover that in the next class. So if you have any doubts regarding the things that we talked about so far, please do let me know. Okay, so this is how we started off. If you have any doubts, please do let me know. Until then, just an intro of the features of Auricle. We have live sessions which are there in Auricle. We have recorded classes which are also available. And there are a comprehensive list of notes which are there for each chapter and subject. And MCQs are also there of different difficulty levels. You can also, if you have difficulties, book a personalized counseling session on their website where you can go one-on-one and clear doubts. And these are the various subscription plans that are currently available. You can use my code Dr. Shakthi 20 for 20% discount on subscription. Okay. And these are the various social media handles. And all of these videos I've seen generally are uploaded to the YouTube channel. So you can always go back in case you have any doubt and see through these videos later. Okay. So if you have any doubts, please do let me know. Okay, so I guess there's no doubt. See you again next week. We'll be talking about diabetes mellitus that time. Okay.